So before I introduce Dr. Donohue, just a couple housekeeping items. As the slide says, if you would please remain on mute at all times. Please note that the session is going to be recorded. And very importantly, please enter all your questions via the chat feature. Dr. Donohue is going to speak. She does not have slides. She's going to speak, and then she will be taking the questions from the chat. So it's very important that your questions go in there. So without waiting anymore, Dr. Donohue, again, welcome. She had joined the graduate of Virginia, I'm not going to go through your whole resume here, but a graduate of Virginia Commonwealth University. Dr. Donohue joined the FDA as a medical officer in 2014 in pulmonary and allergy division. She moved to Inborn Errors Group as a clinical lead in 2017, and then became director of the Division of Rare Diseases and Medical Genetics in 2020. So you've been a popular figure around RDCRN lately, and we're so pleased that you're here. So thank you, and please, the floor is yours.
Kristen, thank you so much. I'm really grateful for the chance to meet with you guys today, and I just sort of want this to be a conversation that is of service to you. So please throw your questions into the chat, and I will do my best to address them. I didn't make slides, but I did kind of map out a couple of things that we can talk through about, what I see as the real opportunities for patient involvement in drug development, and things that, the rare disease patient community can do to really advance therapies. So I'll kind of march through some of that, but like I said, just throw it in the chat, and I'm happy to have this be conversational, and I want to make sure that we're touching on the topics that matter to you. So the points that I organized were sort of by stage of drug development. So starting in the preclinical stage, right before any trial is going to go into human patients. As patient groups are thinking about where to allocate, if they have fundraising dollars or research efforts, I think developing an animal model of the disease is one of the very first steps. So diseases that have a good animal model have a really good shortcut for drug development, and the ones that don't have a much harder time. So if you don't have a good animal model of the disease, that's worth focusing on in terms of research advocacy and fundraising and dollars, because you can expose an animal to candidate drugs and see if you're getting any kind of response. It can help with dose selection to make sure that, we're going to pick a dose that's actually going to have some benefit in humans. It provides this preliminary evidence of efficacy if we want to enroll pediatric patients in early trials. You can use it to get things that drug companies like fast track designation. So, and then you can even use it for confirmatory evidence in an eventual new drug application. So a lot of our drugs in rare diseases get approved based on just one clinical trial, but they have to have what's called confirmatory evidence. And an animal model is a really good source of that confirmatory evidence. So a good one of those is useful for speeding things up at almost every stage of drug development. And so if you have a research organization or fundraising dollars, or you can advocate for it, like getting your community focused on, we need an animal model, that's a good use of research dollars and advocacy. So that was one thing that I wanted to sort of start with. Another really big area of work is biomarker development. So a lot of the genetic diseases that I work for might have a pretty good biomarker, right? And it's usually a diagnostic test. They're looking to see, does the patient, are they missing some normal amount of this thing, or do they have too much of this other thing? And typically what we're doing is comparing patients to, to healthy people. And so we're just looking to see, do they have the diagnosis? Do they have way too much or not nearly enough of this biomarker compared to people who aren't sick? And so that's a diagnostic biomarker. And a lot of our rare diseases have a pretty good diagnostic biomarker. The problem is that that is not a prognostic biomarker. Oh, I've got a question. Thank you, brave questioner. Can you talk a bit more about animal models? We hear most about mouse models. Is that typical? Yeah, so mice are cheap and easy to work with, and you can make a knockout mouse. So if you've got a single gene defect, you can sometimes introduce that into a mouse model, and then you've got an animal model of the disease. Now, it depends. Sometimes some animal models are better than others, right? So some really reliably reproduce the clinical features of the disease, and others don't. And then there are spontaneous animal models. So there are some animals that get the same diseases that the humans do. So if one of those already exists, then you don't have to make it. So just sort of exploring, what your options are. Kristen, do you want to clarify your question any more about that? Did I address that? All right, you can let me know.
Sorry, I was
No.
find the mute button, but yes, you did. Thank you, Katie.
Okay. Okay, next up, so biomarker development. This is a really powerful use for the Rare Disease Clinical Research Network. So a good thing, anytime you've got a biomarker, that also makes drug development a lot easier and faster. So typically we've got one of these diagnostic tests, but they're not like super great for disease prognosis. So you can tell if a patient has a lot of something or not enough of something, but we don't know how much change in that biomarker is going to predict clinical outcomes. That's like the big unknown, and that's the piece you need to really use it for drug development. Okay, here we go. When does a mouse model need to go to monkeys or another animal versus being able to go from mice to humans? So this this is a really hard question, and it very much depends on the disease and the animal. I don't know that I'm going to be able to address that with like general principles. It really matters. The specifics of the science really matter here. So there's animal testing for safety purposes, which is a whole separate topic I'm not going to go into. And then there's what I'm calling animal models for efficacy, for being able to tell, is a drug helping? So for efficacy, if you've got a mouse model or a rat model or a cat model that reliably reproduces the major symptoms of the disease, then you're good. You've got a good animal model for testing efficacy. So you don't need to be going into monkeys if you've got a good animal model in mice. Some of it matters, like which part of the animal is most different from humans, right? So the immune system is one of the things that's the most different. So if you've got a disease with a lot of immune components, then a mouse model may not be very helpful, and you might well need a primate model to reflect that aspect of the physiology. So it depends on how good the animal model is at reproducing the clinical signs and symptoms of the disease. And depending on, is the disease affecting, some core metabolic process that's common to all mammals, in which case a mouse model might be just fine. But if the disease affects a process that's only common to like higher order primates, well, then you might need to be looking at that animal model. So I'm hoping that addresses that piece of that question. Okay, good. Okay. Anything else on animal models before I move back into biomarkers? Animal models are patented. What kind of industry partners are working on these, and who might be best to reach out to? So your academics can be working on these. A lot of these animal models are developed by academics, which is part of why I think it's a good opportunity for discussion between the RDCR and investigators and the patients. So yes, industry does have some of these, but academics can make them too. Patrick, does that address that for you? Okay. Anything else on animals? Okay. Right, so biomarkers. What's my favorite animal? Oh goodness. A zebra. I work in rare diseases. Okay, so for biomarkers, this is a really good area for collaboration between academic investigators and patient groups. So working together on developing, the biomarkers is a good opportunity for patient groups. So why is that? Well, if you have a disease registry that's measuring clinical outcomes and you've got an academic that's working on developing a biomarker, well, maybe you can put those two together, because what we really need to know with a biomarker is how much change in this biomarker predicts how much change in the disease, right? So cholesterol is a good biomarker because we know that amounts above a certain level really increase your risk for heart attacks and strokes and things like that. And so we know how much change there we need in order to prevent bad clinical outcomes. And so that is the missing piece for a lot of these otherwise pretty good diagnostic biomarkers in rare diseases. And it's missing for a reason, right? Like, this is hard work to do. It takes time. You need a lot of longitudinal registry data, and you need a lot of biomarker samples in order to address that question. But if you have a good biomarker, if we know how much change in this biomarker predicts how much change in the clinic, well, now we've made it way easier to pick the right dose for a drug and to run a clinical trial. So a huge part, okay, here we go. How does the FDA relate the biomarker to the patient voice and what is clinically meaningful? Well, this is why it's super great if patients are involved in the work of determining, what that biomarker is measuring. What are the outcomes that matter to you? So would a demonstrative improvement in diagnostic markers be enough to enter into human trials, even if we can't correlate to clinical outcomes? Sure. So I think, Patrick, what you're talking about might be what we call a pharmacodynamic biomarker. So biomarkers can be used for different purposes, just like, you have a hammer in your tool chest and you can use it to do some different things, but you can't use it to do all the things. Some biomarkers have multiple purposes and some have very limited purposes. And it depends on their scientific properties and luck to a certain extent and how much evidence you have. So for sure, biomarkers can help you, even if you don't know, for example, to take Patrick's example about a diagnostic biomarker. Yeah, we often use diagnostic biomarkers as pharmacodynamic biomarkers. So we're willing to tolerate a pretty high degree of uncertainty in a pharmacodynamic one. And we use those to pick drug dosages and, as Patrick noted, to identify potentially relevant candidate drugs for moving into the clinic. So yeah, the level of evidence needed for a pharmacodynamic biomarker is a little bit lower than the level of evidence needed for a prognostic biomarker, but they're useful even so. Because one of the big reasons why rare disease drug trials fail is we didn't do enough dose ranging, and it's easier to do good dose ranging if you've got a good pharmacodynamic biomarker. But that biomarker development, can we use it for diagnosis? Can we use it to pick a right drug dose or to identify potentially promising therapies? Can we use it as the endpoint for our trial? So one of the reasons why we have so many more cancer drugs on the market than we do for other rare diseases is because they've developed really good biomarkers. They had decades of scientific work identifying those biomarkers, and now they have a pretty good sense, if we have this much change in our biomarker, then we know we're going to have this much change in the clinic. Okay, Penny has a question. What are the different tactics to work directly or indirectly with the FDA regarding the conduct of clinical trials and resulting decision-making? Which tactics are in the pre-competitive space versus advocating for a specific company's medication and development? Oh, Penny, that's the $64,000 question. Okay, let's go there. So I think there's a bit of a misconception about when and where patient involvement happens most effectively. So one of the challenges for us is, obviously patient preference and patient voice matter. So I would love to see way more patient involvement early in development. So in this preclinical stage and in the design of the clinical trials, right? Like, patients should be the ones helping us pick endpoints and helping us design a trial that's going to minimize burdens and maximize the chances of finding an effective therapy. Like, I really want to hear what patients think as we're doing that. And there's a balance, right? Like, some things that measure or that matter a lot to patients are really hard to measure scientifically. So that's not going to be a good endpoint, but we need to find something in that Venn diagram that matters to patients and that we can measure reasonably well. You need both. And so obviously patients have a piece of that puzzle. So I would love to see a lot more patient involvement early on in the trial design and endpoint selection phase. In rare diseases, the other place where you can have, where I think patient preference and patient voice can play a really important role, is if you have a lot of approved therapies, which unfortunately for most rare diseases, it's just not the case. But let's say you're one of the small groups of rare diseases that has several approved therapies. If you've got another therapy coming online for marketing review, so it's submitted to the FDA as a new drug application, and maybe it has a novel safety profile. It has a rare but serious risk that's different from some of the other drugs, but it also has a little bit of a benefit that's different from some of the other drugs. So that's a place where I think patient preference can play a real role in terms of informing the kinds of risks and trade-offs that you're willing to accept. Where I think there's a misconception is how much of a role patient preference can play when we're talking about a drug and there's no other approved drugs. So every patient would prefer to have an approved treatment. I would, all of us would. But a patient preferring to have a treatment that's FDA approved is not the same thing as having scientific evidence that the drug works. And so the job of the FDA scientists really is to look at that scientific evidence and determine whether or not we're seeing what we call substantial evidence of efficacy. It's fundamentally a scientific question. It's our core responsibility, and it's an area where, you know, that's a scientific process. And so we know patients would prefer to have an approved drug, but a patient preference for an approved drug can't really substitute for that substantial scientific evidence. And so I think it's on us at the FDA to be clearer in talking with patients about where in the process their voice can have a big impact and where there are some limits on it. And so I think it's important for patients to understand that advocating for a specific drug may not be as helpful as companies are telling you it might be, or some patients might want it to be. I mean, I think we just need to be honest about, you know, what can that patient advocacy do and what can't it do? You know, you want to bring a hammer to the right job. You know, if you're hammering a nail, you should use a hammer. If you're trying to solve a scientific problem, well, then we need scientific evidence. So Penny, does that address your question? This is a big topic, so I want to make sure that I circle back to you and see if you have anything else.
Can I just unmute, or it's a lot faster?
There you are. Now I hear you.
No, that was really helpful, because I think that we often get confused sometimes thinking about the ways that we interact with the FDA, that it's not just about, like, there's the places that are appropriate and there are the places that are less appropriate or not at all appropriate. I wonder if you could talk a little bit more about some of the other approaches, like there's things like the research roundtables that some disease spaces do, places to have pre-competitive discussions that are science-based, but where a nonprofit advocacy organization can be a convener for those types of scientific discussions.
Absolutely. Yeah, so I mean, the animal model, the biomarker, and this is, I mean, there are patient groups that are really advancing this. So I think those are areas that are really ripe for focusing advocacy efforts. And maybe it's advocacy at NIH, like, hey, you know, we want you to put research dollars towards animal model development for this family of diseases. Or we want you to put research dollars towards biomarker development. So I think there's a role there for advocacy in terms of understanding what are the gaps in your disease pipeline in terms of drug development. So if you don't have an animal model, I would start there. If you don't have a biomarker, I would be advocating for research dollars to go there, whether that's a combination of public or private funds. Digital disease monitoring is another one that I think is really ripe for registry involvement. So, you know, we're just beginning to, and I specifically was asking Penny to chime in. So Kristen's trying to keep us all in the chat, but I really did want to hear from you, Penny. So that's on me. So digital endpoints, right? There's all this excitement about maybe we could use iPhone apps to monitor ambulation instead of a once a year 6-minute walk test, or maybe we could use, you know, there's actually an iPhone app you can use to measure ataxia. These are all like really brand new tools. We don't understand a ton about how they work, but I'd love to see patient registries using and collecting more of this data longitudinally so that we can start to learn and see, well, when and how can we use those digital endpoints instead of, you know, on-site study visits to measure that kind of thing. Because I think it has a lot of potential to minimize burden for patients, to allow decentralized virtual study visits. I think there's huge promise there, but we have a lot of work to do. And that's one where data from across the community could be helpful, right? So if we have lots of patients with rare diseases measuring the same kinds of outcomes, using the same kinds of apps to measure walking or ataxia or whatever it is, speech, and we're starting to see and get tons and tons of data in a variety of disease conditions at a variety of ages and in a variety of settings and a variety of devices, I think that's really going to help advance some of this endpoint development for very rare diseases. So I would love to see patient groups incorporating more of those sort of digital endpoints in their registry data collection, because those might be better endpoints. Another area that's really great for patient involvement in the preclinical space is guidelines. So there's a couple things here. One, when we want to put pediatric patients in a trial, we have to be able to measure the endpoints. So let's say, so this is one of the areas where cancer has a huge advantage over a lot of other rare diseases. So cancer clinical guidelines require imaging at regular intervals. You got to measure tumor size every so many months, even in pediatric patients. And it's part of the clinical guidelines. And so if we need to run a clinical trial in pediatric patients with cancer, well, the radiation associated with the CT scan is not considered an added risk, even in patients in the placebo arm, because they would be getting that scan anyway as part of their clinical care. So anytime you can take your potential clinical trial endpoints and make it part of the disease guidelines, part of the routine monitoring and management of the disease, that makes it a lot easier for us to require that as an endpoint in our clinical trials. So as a community sort of converges around what makes sense as an endpoint. So maybe it's a serum biomarker that was initially developed as a diagnostic test, but the community's done all the registry work to show that X change in this biomarker causes Y change in clinical outcomes. They've done all that work in the registry. Well, make measurement of that biomarker part of the clinical management of the disease. We're going to use that biomarker to predict clinical course. We're going to use that biomarker to determine when patients might need to move to additional respiratory support or ambulatory support or feeding tube placement, right? So anytime you can incorporate that endpoint into the routine clinical monitoring and management of the disease in your guidelines, it's going to make it a lot easier to do the clinical trials we need to do, because all the doctors will already be used to measuring that thing. They'll know how to do it. They'll know what it means. Patients will be familiar with it, and you'll have a whole lot more natural history data around that endpoint than if it's not. Okay. So that's clinical guidelines. So it has a lot of benefits. If you've got any kind of biomarker or potential endpoint that is often measured, maybe think about asking your academics to put it in the clinical guidelines so that everybody's getting it measured regularly, and then pull that data into your patient registry. And then the last piece, we're starting to see a couple of patient groups, some of the more established ones, start to launch platform trials. That's a big lift. Small patient groups that have never even had a therapy in the pipeline are not going to be ready for this. But I want to put it out there as aspirational. So the FDA loves platform trials. Patients love platform trials. Drug companies hate them. And that's why they mostly don't happen, is that drug companies hate them. So what is a platform trial? Platform trial is your patient group can functionally run the infrastructure for your clinical trials. And it means that when a company has a drug they want to develop, they come to you about, can we get it into your platform trial? So some of the cancer drugs work this way. Duchenne muscular dystrophy is doing this. And the real advantage is it minimizes the number of patients who need to be in placebo. And it also allows for pretty quick determinations. If a drug isn't working, then you take folks off that arm and re-randomize them to something else that might work better. So one of the challenges that we see in a lot of drug development programs is that once a patient chooses, if you've got a diagnosis, lucky enough to have 2 or 3 candidates in development, it's not uncommon. Once patients choose to try one experimental therapy, they end up staying on it long-term, open label, and then that blocks them then from participating in trials for another therapeutic, even if there isn't particularly good evidence for whatever it is that they're on. So the advantage of a platform trial is you've got a steering committee and you've got patients involved and at the table. And if a therapy fails, there's this opportunity to stop and rapidly reassign those patients to other trial arms that might have more potential. So I think there's and in the overall, you need far fewer patients on placebo in a platform trial, because really what it is a shared placebo arm. So instead of having one standalone trial where patients get randomized to treatment or placebo, you might have 3 candidates in the pipeline. And so patients might get randomized to placebo or drug A or drug B or drug C. And then if it turns out drug B is not working at all, then those patients might get re-randomized to A or C or placebo. And so instead of having half of the patients in any clinical trial in placebo, you can have 3/4 of patients on treatment and only 1/4 on placebo. So there are real advantage to patient communities in terms of minimizing placebo exposure and minimizing exposure to ineffective therapies and sort of rapidly reallocating patients. So which organizations are running these? Yeah, Duchenne muscular dystrophy is probably one of the better examples. And then there was also a breast cancer platform trial that you guys could look at. Obviously not a rare disease, but in terms of what it looks like when an advocacy organization stands one of these up. It's resource intensive. It's going to be a heavy lift for smaller organizations. I suspect that the way we're going to get there is probably a broader, like, rare disease platform trial. If I could wave a magic wand and get Congress to do anything at all, I would ask them to stand up that infrastructure for patients with rare diseases, like sort of a national rare disease clinical trials infrastructure. But I just want to put it on the radar screen as something to think about if you're maybe a slightly larger patient group, or if maybe you could partner with related diseases so that you've got 10 centers of excellence that are recruiting patients with 10 different diagnoses into this common infrastructure. There might be some opportunities there. Okay, so that's guidelines and platform trials. So to recap, things that patient groups can do to advance drug development in the preclinical stage: get an animal model, develop your biomarkers, and that can be a serum test. It could be imaging, just anything that predicts a clinical outcome instead of having to wait to measure a clinical outcome. Digital endpoints, clinical guidelines, make whatever it is that you're measuring, your possible endpoints, like incorporate them into clinical guidelines anytime you can. And think about maybe trying to launch a platform trial or to partner with some related patient groups about doing that. So that's preclinical. How does a platform trial differ from a comparative trial? So this is one of the reasons why companies don't like platform trials, is that they give you comparative efficacy data. So if you do a standalone trial and you try drug A against placebo, and you do a different trial and you try drug B against placebo, you can't directly compare drug A and drug B. And pharmaceutical companies like that. And that's one of the big things they hate about a platform trial, is you get comparative efficacy because you're all comparing to the same placebo. And so FDA loves these trials, patients love these trials, but for obvious reasons, pharmaceutical companies don't. And they have other reasons why they don't love these trials too. You're trusting other companies to collect some of your data for you. And so, some companies are better at that than others, and they might not want to take that risk. So there are some legitimate business reasons why pharmaceutical companies might not want to do that. What would best persuade pharma to do a platform trial even though they don't like them? Well, if you build it for them, they will come. And frankly, you will have done a lot of the work for them. So yeah, patient access, exactly, Patrick. So if you as an organization are really in the lead when it comes to recruiting patients into a clinical trial, and you've got all your patients already enrolled in a platform trial that's ready to go as soon as a therapy comes online, you've already got patients used to doing an annual visit or whatever the remote study visit is to measure the biomarkers or the disease endpoints or whatever it's going to be. You're collecting that natural history. Well, as soon as, if you've got that infrastructure in place, as soon as a therapeutic comes on board, you can say, yeah, we can fully enroll your clinical trial tomorrow. Put it on our platform. And you're going to randomize patients to placebo and to your drug. And if we get another candidate in the pipeline, you're going to share your placebo data with that other company, and they're going to share some of their placebo data with you. Like, those are the rules. Oh, okay, I've got another. Oh yeah, Ed. Ed, do you want to just chime in? Go ahead and share that verbally.
Hey, Katie. This is a great session. It really is. I don't need to take much time. I just thought it might help the group to hear a few more examples of platform trials. And one that came to my mind was the one that's being coordinated by the Healy ALS Center at Mass General.
Mm-hmm.
They're active at many other sites. And it's not coordinated by a drug company, as you said. They don't like them, but coordinated by an academic institution.
Yeah, and even though I'm saying they don't like them, they are working well. And I think pharma companies have seen there was a lot more with platform trials during the COVID-19 pandemic. And I think folks are starting to get more comfortable with the idea that like they really can work and they can work really well. And they have the potential to be a win-win for pharma companies too. So I think if you've got a community that's already organized and prepared to do that, I really think it can be like a win-win-win for patients and academics and pharma companies. But the pharma companies are probably not going to do it by themselves, I guess.
I would agree with that. And once the platform trial is set up, especially for smaller companies, that would be their path of least resistance to get involved rather than to try to set up something completely separate.
Exactly. That's exactly right. Okay. So moving to the next stage of drug development and what patients can do, which is as you're moving into the clinic. So this is trial design. And one of the designs that we are recommending all the time is what's called an adaptive design. Now, historically, the way people would do this is what's called sample size re-estimation, which is a fancy pants term, but it basically just means you check halfway through your trial and see how it's going. And if it's not going as well as you'd hoped, then you add a bunch more patients into your trial so that hopefully you can still win. Now, obviously, that does not work in rare diseases because you've already put all your patients in the trial. But instead of adding more patients, what we can do in rare diseases is add more time. There is a scientifically sound way of doing this, and you have to agree ahead of time. We are going to have a select group of people who will get to look at the data at these pre-specified times. They're going to look at 3 months, 6 months, a year, 2 years, whatever it is. You decide ahead of time. This carefully selected group of people is going to get to look at the data and decide what to do. And the other thing you can do is decide your futility and success criteria. So let's say you've got your platform trial up and running, you've got 3 candidates in the pipeline. Patients should be deciding, well, if we're not seeing any difference at all by this time in our endpoint, we don't want to keep our patients and kids and loved ones randomized to something that's not working. So, what's your futility criteria? As a patient, when you say, I'd rather be able to switch to another trial arm where I've got something that looks a little more promising. So if we're, not seeing any difference at all at 9 months, I want a chance to get out of that and into a different trial arm, right? So patients should absolutely be defining the futility criteria. And patients can define the success criteria. So if we get a treatment that's just gangbusters effective, like, wow, this stuff is amazing, maybe we can stop the trial a little bit sooner. But if we're seeing modest benefit in the treatment arm, but it's a little smaller than we'd hoped, but it seems to be there, well, maybe folks would willing to stay randomized for a little bit longer in order to demonstrate benefit and get that drug approved, right? A lot of our drugs are modestly effective, and so longer trials might be necessary. But that means patients have to be willing to stay randomized longer to detect modest drug effects. And so how long are you willing to stay randomized? If you want drugs approved with modest benefits, as a community, you might need to decide we're going to have to be willing to stay randomized a little bit longer in order to get a drug with a modest benefit approved. So I think patients have a real role to play in defining futility and success criteria, and defining how long they're willing to stay randomized in order to detect more modest benefits. Obviously, endpoint selection, like patients are integral to that. We touched on it before. Let's see. And then, patient-reported outcomes. So I want to touch on that because I feel like FDA does a lot of talking about the role of the patient in patient-reported outcomes, and we like them because they're measuring directly what's happening to patients. And I just want to say a word about that. I think they work really well in some scenarios, and they don't work at all in others. So if you have a disease with frequent daily symptoms that like wax and wane, you know, like we've got a recent example of like itch, where there's a disease that causes horrifying itching most days. And so if you've got a drug that's highly effective at controlling itching, then asking patients, how itchy are you today, is probably a pretty good endpoint, because it's a pervasive, prevalent symptom, and it's potentially reversible with treatment. So if you've got frequent, like waxing, waning, reversible symptoms, then a PRO can be a good endpoint. If you've got one of the diseases that I work on, like a slowly progressive neurologic disease, run away from PRO endpoints. It's like a one-way ticket to failure. Like it's so hard to detect a slowing of progression in disease on a PRO endpoint for neurodegeneration. Like almost none of our drugs are going to reverse that. And so you're really looking to detect a slowing of progression, and that is hard to see on a PRO endpoint. So thinking about the nature of your disease and what you can measure. So like, you know, cognitive function is very, very important to patients, but all of our tools for measuring it are terrible. We have no good way scientifically to measure it in small patient sizes. So we need to be thinking about biomarkers and other kinds of endpoints that we can measure more easily. So endpoint selection, that balance between what patients care about and what makes sense scientifically, and like really working with your scientific advisory boards and your academics to kind of understand that. And patient groups have a role to play in educating their membership about that, that like, yeah, we know that what we care about is cognitive function, but we can't measure that well. And so we need to pick something else that maybe isn't as important to us, but that we can measure so that we can actually get a clinical trial done. It's that practical piece. And I think patient groups have a big role to play in kind of educating their membership. Have I seen anything with measuring swallowing? Yeah, so video studies are good. Any endpoint that directly measures function is going to be pretty good. So lung function is a great endpoint. You know, a video swallowing study where you're watching the patient swallow and you're able to quantify the degree of aspiration. You know, any endpoint like that. 9-hole peg test is good because it's a direct quantification of fine motor. So any direct functional test is going to be a pretty good endpoint, and a pretty good endpoint in a global trial, right? One of the other challenges with a PRO is that most of our rare disease trials are global. You're pulling patients in from a dozen countries that speak a dozen different languages. A lot of the PROs are very language specific. They're very context specific. You know, if like I've seen PROs that ask questions about education, like an IEP plan. Well, not every country has IEP plans. And so if your endpoint is asking about them, it's not going to be relevant in countries that don't do that. Whereas a functional test, like the 6-minute walk test, is a 6-minute walk test anywhere in the world. So functional tests are often a lot easier to do in a global setting, whereas PROs can require a lot of work to translate and be useful. And they don't always make the leap, is the other risk with a PRO. Okay, let's see. Oh, and then these decentralized, pragmatic, remote study visits, I'd love to see more of these just to minimize burden to patients. So let's see, right? So there's a big move to take clinical trials to hybrid trials, like in-person and remote. Are there guidelines for doing a reliability assessment tool for rare disorder to make remote visits possible for patients and decrease burden? Yeah, Jody, so this is what I was touching on earlier about incorporating digital endpoints in your registry work and observational trials. So if you can show that, you know, let's say you've got an annual in-person visit for 6-minute walk, and you're using some pedometer app on an iPhone that, that you keep with you. If a community can show that these things correlate, and that actually of the two of them, it looks like the iPhone app is the better measurement because you're getting continuous longitudinal data instead of just one snapshot a year. I mean, that's really valuable, but you've got to be able to show that. So we know how 6-minute walk works and doesn't work. We don't know yet how the iPhone apps work. So you've got to collect a whole lot of data to get a feel for, you know, what degree of change on the iPhone app is going to be clinically meaningful. So we know what that is on 6-minute walk, but we don't know yet on an iPhone app. So we just need lots of people to collect it. But I think it's that registry natural history piece across a range of diseases that we need to collect in order to have more of these trials remote and virtual and fewer, travels for in-person study visits. I mean, I would love for every single rare disease clinical trial to send a nurse to your house instead of you having to come to an academic medical center. I just think it would be better for patients and we could enroll more patients in the trial. And do we have guidelines for this? The core question is really scientific. So I don't know that we have, I think there are some guidelines in development. I don't know that they're published about digital endpoints in particular. But the core issue is really scientific. It's like how you would develop any other endpoint. And so a lot of the guidance on PRO development or endpoint development would be pertinent. It's just now it's digital. Okay. Right. Okay, this is a great question from Dominique. So if PROs are not used, how does the FDA assess that an objective improvement, say improvement in 6-minute walk or swallow, is meaningful to the population tested? That's a great question. So for the 6-minute walk, a lot of the initial data were actually collected in COPD patients, and they found that about 30 meters difference correlated with a difference in their clinical outcomes. And then they found that 30 meters seemed to be sort of a magic number. It was true in a couple other disease settings too. And so once you start to see that a certain amount of change on a certain endpoint is relevant across a range of settings, you have more confidence in extrapolating that in a rare disease. So we've used 6-minute walk as an endpoint for several rare diseases without necessarily having a ton of data in that specific disease for us to know. Right, so on a certain degree, we're extrapolating. Same thing is true with lung function, right? So there's a certain degree of change in lung function that we know correlates with symptoms in COPD or asthma. There's a certain degree of lung function that we know actually predicts mortality, even in healthy volunteers. Same thing in patients with interstitial lung disease. So we've got a sense of how much change in lung function is likely to matter across a huge number and a huge range of patients. And so that gives us a pretty good guess that this amount of change in lung function might also be meaningful to patients with a very rare disease, even if we have a much more limited data set in that rare disease. So that's part of what I'm talking about when it comes to determining what amount of change is clinically relevant. It's typically a mix of disease-specific data and then some measure of extrapolation, especially when you're talking about rare diseases. We're never going to get enough data in that rare disease probably to know for sure. And so there's a little bit of extrapolation going on. So if we can see that X amount of change is relevant across a wide age of patients, range of diagnoses, that gives us more confidence that we're looking at something sort of physiologic and fundamental that's going to translate across disease states. Dominique, did that address your question? And let me see what else we've got up here. What are the mechanisms where patients are able to define futility and success criteria? PFDD meetings, advisory role in specific clinical trial designs. Yeah, Penny, that's exactly right. So I would love to see patient groups. I mean, you guys could do the work of designing your own clinical trial at a PFDD meeting. Like, find out how long are patients willing to stay randomized. There's a direct trade-off between being able to detect modestly effective drugs and how long folks are willing to stay randomized. And so if you can find that and educate the community and help them understand that, and you can get a community where you've got 80% of patients say they're willing to stay randomized for X amount of time in order to detect Y amount of treatment effect in a randomized trial, you have positioned yourself really well to partner with a pharmaceutical company. Like, you guys have already done the hard work of getting your community on the same page about what a clinical trial might need to look like. So communities that struggle are often the ones where they come to the table and say, oh, nobody's willing to be randomized. Well, that's like telling a contractor you want them to build your house, but you don't want them to use a hammer. Like, okay, how do we develop drugs? Like, most of the time it's randomized trials. And so if a community is coming to the table saying, like, we want you to develop this drug, but none of us is willing to be randomized, well, that's hard. So a lot of what communities can do is really support and educate and help their members come together and reach agreement on what is a randomized clinical trial that we could get behind. Let's see. Okay, Paul is asking a question about, would you give more detail on my earlier comment about cancer having more therapies because they have better biomarkers? Is this accelerated approval or is this earlier in development? Paul, it's all the way across the board. So those biomarkers are really helpful for for you know, diagnosis, for clinical trial entry criteria, for detecting potentially promising therapeutic candidates, for picking out the dose. A good biomarker can accelerate drug development every step of the way, and then, yeah, potentially be used for accelerated approval. The other real advantage in the cancer space is that, for better and for worse, cancer kills patients really quickly, which is terrible in real life, but it has scientifically useful properties, right? So it's a little easier if all the patients are still alive after a year on a new treatment, and we would have expected them all to have died by then if they had metastatic disease. Well, it's really easy to show when you've got a highly effective therapy. And so there's not a big gap in time between an accelerated approval on the biomarker and when you get your clinical outcomes results. So for rapidly progressive diseases, frankly, they're easier to study. So the more severe diseases that I've got in my portfolio, they're easier to study. You get your clinical outcomes and your results in your trial a lot faster. The slowly progressive diseases are just harder to study because it takes longer for you to figure out whether or not the drug is working. And so some of the difference in, you know, which drug development programs take longer has a lot to do with the underlying disease. So the cancers move pretty quickly, and so do their drug development programs. Some rare diseases progress really quickly, and their drug development programs can progress quickly too. If you've got a slowly progressive disease, they're just always harder to study. It takes longer. And if you don't have a good biomarker, it can take a really long time. So if you've got a slowly progressive disease, figuring out a biomarker you can measure sooner is going to help everything. Yeah, and Dominique's noting that it's really challenging in neurodevelopment disorders that don't have any objective measure used in more common disorders. I totally agree. But that's why I wonder if like an iPhone app for ataxia might be a game changer here. Or, you know, neuroimaging, right? Is there work we can do on neuroimaging that would be helpful? So I'm right there with you. Finding good endpoints for neurodevelopmental diseases is a huge challenge. And the other piece we deal with is that there's this like push-pull in the slowly progressive ones in pediatric patients, where the kids might have some normal neurodevelopmental progress, and then they might suffer a setback, and then they might have a little bit more, and then back. It makes it so hard to study. Okay, we have 10 minutes. I've covered a lot of what I wanted. And as we've been talking about this, and Penny, your question really zeroed in on this, is like, well, what about a marketing application? So companies will often come and tell patients like, oh, you know, we want you to go have a listening session with the FDA about our drug. And I have to tell you that unless there's like a really unique safety trade-off relative to other approved therapies, like that's a use case where I think patient voice can have a really important thing to offer at that stage. But if we're talking about a disease with no approved therapies or very few approved therapies, the real question at that point from a marketing standpoint is typically, does the drug work? And that's a scientific question, not a patient preference question. So let's see. And just, you know, my division right now is getting a ton of asks for patient engagement during a marketing application, more than we can say yes to. And we love meeting with patients. I love talking with patients. It's breaking my heart to have to turn some of these down. But right now we're getting more requests than we can say yes to. And so I'm really prioritizing the ones that are sort of earlier in the pipeline. Like, where can we have a really good scientific question? Where can patient input like really make a difference? Let's put our time there. So that's really what we're focusing on. And I'm having to say no to some of the asks when we've got a marketing application in-house for a specific drug and there's no other approved therapies. I'm like, this is a science problem. We've got to deal with that as a science problem. Okay, more questions. Let's see. Okay, questions about rare disease clinical trial infrastructure. Yeah, I mean, the FDA does not, that's like a moonshot kind of expense. So no, FDA does not have money to stand up its own clinical trials infrastructure right now. And I'm not aware of plans to do so. I think we were more just sort of curious, like, what is the state of play in terms of rare disease? So that's what that RFI was about. That's what Penny was asking me about. Raj tells me I've got 10 minutes. Looks like it's 8. Do you guys have any more questions for me? Okay, animal models and biomarkers. How would the FDA view HIPSCs and organoids as models for preclinical studies? I mean, we're interested in anything that works, right? So if you've got an organoid that really predicts potentially effective drugs, I mean, that's great. That's even easier than using an animal model. So there's a variety of ways that you can screen for potentially therapeutic effective drugs. And the easier and cheaper and faster it is to do that, the more candidates you're going to be able to screen. What else? You guys are asking great questions. This is fantastic. Where are we with cellular therapies? Okay, this has like patient-specific info, so I'm not going to go too much into it. Right. So gene therapy is like the huge exciting thing, and I think it has so much potential, and I think we're on the cusp of some real game changers. It's early days yet, but I think there's a lot of potential, and you know they present some really different challenges from sort of conventional drug development. Probably more so than I can get into here, but honestly, like I think it's really exciting, and that might be a great topic for like another whole patient advisory group meeting. What else? We give an example of a best case scenario with patient, community, academic, and industry. Oh, I have lots of them. How they worked well together. Yeah. So let's see. An example of best case scenario and how they work well together. So I think patient groups really are central to that. So patient groups have a ton of power. Your ability to get a clinical trial fully enrolled is a huge asset to bring to the table for a pharmaceutical company. The hard work the community has done to identify biomarkers or an animal model or a clinical trial design that's broadly acceptable scientifically and ethically into the community, that sort of work on cohesion. Patient groups that have a good cohesive community that together is committed to advancing the science and to the hard work that it takes to advance the science. It's really hard work. And if you've got a really fractured community and a whole lot of people who aren't willing to participate in the science and in controlled trials, it'd be really hard to get drugs developed. So that work around cohesion, around doing the hard work of good science, I think is where patient communities really sink or swim. And it's the stuff that most of you guys are already doing, right? If you're interested enough to listen to me blather on for an hour, you're probably already doing this, right? Okay, let's see. So Raj is asking me, okay, this is the last question. The FDA has become very good at listening to patients and families. Can you share how that listening is incorporated into what is a very evidence-based review and approval process? Thank you. Yeah. So if you've got a voice of the patient report, we quote those all the time. So if I've got a company, like, let's see, last year I had a company that wanted to measure, they had a drug that they thought worked a specific way, and they wanted to measure this endpoint that they thought they could get like a quick win on, which, fine, I'm not opposed to that. But the thing is, it didn't matter to patients. Like this was not a thing that really mattered to patients. And so I was able to go to the voice of the patient report and say, it's nice that you've got a drug that treats something, but like the patients don't care about this. It's not important to them. Like, how can you provide some evidence that your drug is going to make a difference to what actually matters to these patients? Like, can you bring some patients into your next meeting? Can you provide some additional justification? Can we broaden our look for endpoints that matter? Like, what if you redesign the trial and looked at this because it looks like the mechanism of action of your drug might actually move this, and this is something that matters to patients? Can we also measure that? So a voice of the patient report, we read those, we reference them frequently. In fact, I've got a couple diseases where I cite that voice of the patient report like almost every time I talk to a pharmaceutical company. So I think those are directly affect how we do our day-to-day work. So I would say that's probably your best bet, is that voice of the patient report. Another time is in the concomitant therapies. So I had a community where off-label use of a drug was like nearly ubiquitous. And the sponsor was telling me that they were gonna prohibit use of that drug, that concomitant medication, in their trial. I said, you can't do that. You can't do that. Everybody's already using it. You're not going to be able to enroll your trial. They're not going to want to come off of it. Like, you're going to need to develop your new drug as an add-on therapy to that. And it was helpful to be able to go to that voice of the patient report and just show them, like, look, they're all already on it. And then they agreed. So talking about that background therapy has been really important. Let's see. Okay, I think that is about all that we have time for, but I just want to thank everybody for amazing questions. I really appreciate it. Thank you for having me. And, I think you guys have more power and more of a role to play in the science, advancing the science of treating your diseases. It's a lot of work, but it's also a lot of opportunity. So thanks for having me.
Wonderful. Dr. Donohue, thank you so very much. That hour went by like that. I don't think anyone would describe it as blathering on at all. And we so appreciate your time and your talent and expertise, and very particularly for working on rare disease on behalf of so many patients. You are one of our heroes for all the work that
No. You guys do all the hard work. This was really my message. The FDA doesn't do very much. It's you guys who have to do the heavy lifting, but we try to support you as much as we can. So thank you for having me.